So next stage of the prophase 1 is Diplotine stage, that is after which stage? Before that we were studying about the which stage? That was the पैकेटीन स्टेज. पैकेटीन स्टेज के बाद कौन सी स्टेज आती है? डिप्लोटीन स्टेज. Here the synaptonemal complex that was formed earlier, that will get dissolved, and the bivalent or the tetrad that was formed, that bivalent chromosomes, they get separate from each other except at the site of crossing over. So if we see the diagram over here, the X-shaped structures they are formed, which are called as chiasmatas. Okay, so here I have mentioned in the diagram this X-like structure, this one and this here, where there is recombination nodule formation. So these X structures, these X-like structures, you can see here, I have darkened that area in the diagram. That area is called as chiasmata. Clear? So in Diplotine, the characteristic feature is dissolution of synaptonemal complex and formation of chiasmata. Now we come on to the last stage of prophase 1, that is the fifth stage, where it is characterized that terminalization of chiasmata. Now chiasmata will end. Chromosomes, they are fully condensed. Meiotic spindle is assembled for the separation of homologous chromosomes, and finally the nucleolus will disappear, nuclear envelope, it will break down. So by this stage prophase 1 is completed. Now we come on to the next stage after the prophase. We all know, as we have studied in the mitosis, the next stage is metaphase. Okay, so metaphase 1 of meiosis 1, it will undergo the different kind of changes as the bivalent chromosomes, okay, that were formed during the prophase 1, they all will align on the equatorial plate. I have mentioned the bivalent chromosomes on the equatorial plate in this diagram. And what happened? The microtubules, they are formed on the opposite poles and they will attach the spindle fiber to the homologous chromosomes for the movement of these homologous chromosomes to the different opposite poles. Now the next stage after metaphase is anaphase 1. Here in this anaphase 1, homologous chromosomes they separate, while the chromatids they remain associated at their सेंट्रोमेयर. जो इसके सिस्टर क्रोमैटिड्स हैं वो सेंट्रोमेयर से जुड़े रहेंगे. आप देख रहे हो कि दोनों होमोलोगस क्रोमोसोम्स दे विल गेट सेपरेट. द इक्वेटोरियल प्लेट के जो ऊपर थे वो ऊपर वाले पोल पर चले गए और जो नीचे थे वो नीचे वाले पोल पर आ गए हैं. सो बोथ they are separated. Now they have moved from equator to the pole region. Now we will move on to the next, that is the last stage of meiosis 1, that is telophase 1. Now here in the telophase 1, what happened? The nuclear membrane and nucleolus they get reappear, which was disappeared in prophase 1, they will again reappear, and cytokinesis it occurs and form the dyad of the cell. Dyad of the cell means two set of cells they are formed at the end of telophase. After then cytokinesis takes place and at the end two sets they are formed. Okay, so this is mentioned by telophase 1. We can see two set of cells they are formed at the end of meiosis 1. Okay, now here comes the stage called as interkinesis. Interkinesis, it is the stage between the two meiotic division. I have represented it like this: meiosis 1, interkinesis, and then meiosis 2. This stage interkinesis is short lived. Okay, it just occur for very short duration. Now after that we have completed with meiosis 1. Now what will happen? Now the second division it will take place, that is meiosis 2 division. Okay, now this meiosis 2 is almost same like the mitosis process. Clear? So again it is divided further into prophase 2, metaphase 2 anaphase 2 and telophase 2. Clear? So prophase 2 here it resemble normal mitosis process. I have discussed with you, nuclear membrane will disappear and chromosomes, they will become compact. So you can see the diagram which is elaborating, which is depicting the prophase 2 process. Then comes the metaphase 2. The chromosome, they align themselves at the equator spindle attached to the kinetochores of sister chromatids. Same it was happening in the mitosis. We can see that chromosomes, they have arranged themselves onto the equatorial plate, and spindle fibers, they are attached with the kinetochores. Okay. Next comes the anaphase 2, splitting of centromere of each chromosome, allowing them to move towards the opposite poles of the cell. Now you can see in the diagram that centromeres, they get split. Now the chromosomes, they are moving to the opposite poles. Homologous chromosomes, it gets split up, clear, to the different opposite poles. Now then comes the telophase. Two groups of chromosome once again get enclosed by the nuclear envelope. So two set of chromosomes, they will get enclosed by the nuclear envelope, and this stage is called as telophase 2 stage. Now after telophase, cytokinesis is follows, which result in the formation of 4 cells, and 4 of them, they are now haploid daughter cells. As I have already discussed with you in the starting, that mitosis and meiosis, they are different, because at the end of mitosis only 2 cells they are formed, and both of them, they were diploid. But in the case of meiosis 2, 4 cells they are formed, and 4 of them, they are haploid. So this is the difference between these two, meiosis 1 and meiosis 2 and mitosis. Clear? So this was all about meiosis 1 and meiosis 2. I hope you have understood this division process. Clear? Now we come to the significance of meiosis. Now we all know that for the conservation of specific chromosome number of each species, only meiosis is responsible in the sexual reproduction. Now what happened, that this meiosis, it helps in increasing the genetic variability. Why it is leading to the genetic variability? Why it is increasing genetic variability? Because here in this meiosis, crossing over takes place, recombination takes place, new genes, they are able to join with the different kind of genes. So different combinations, they can be formed easily, so that will lead to the variation. And finally, variation, it is important for the process of evolution. So evolution का जो base है वो variation है, variation का जो base है वो crossing over है. Okay, and crossing over is the part of reductional division, that is meiosis. So by this we have completed this topic as well as this chapter, which was including the cell cycle and cell division, mitosis as well as meiosis. I hope you have got the all of the processes, you understood them well, and if in case you have any doubt, you may discuss with me. Thank you.